Welcome guys, welcome to the another video. In this video we will talk about the urine formation. In previous video I talked about the urinary system, but in this video we will talk about the urine formation. So stay with me and know about this topic. We will talk about briefly about the urine formation, how actually the urine is formed inside the kidney. Here I made the structure of the kidney. In a previous video I also talked about the kidney. As we know, the kidney, inside the kidney there are present two regions. The first one is cortex and the next one is medulla. But while the nephron is present inside the kidney, so the first this region it's called cortex. It's called cortex region, and this region it's called medulla. So while the nephron is present, this is the structure of the nephron. It's here I made the structure of the nephron. The first part of the nephron, it's called Bowman's capsule. Okay, the Bowman's capsule is located in cortex region of the kidney. So this is the Bowman's capsule. This cup-like structure, it's called Bowman's capsule. This Bowman's capsule is present in cortex region of the kidney. So this is the Bowman's capsule. This cup-like structure, it is located in cortex region. After the Bowman's capsule, there are some tunnels. These tunnels are called PCT, proximal convoluted tubule. This PCT part of the nephron, it's present also in cortex region. This is cortex region, and this is medulla. So the Bowman's capsule, it is present in cortex region of the kidney, and the PCT, okay, this PCT part, this PCT part of the nephron, it's also present in cortex region of the kidney. After the PCT, there are another structure. It's U-like shape. This U-like shaped structure, it's called loop of Henle. This loop of Henle, it's present in medulla region. This loop of Henle, this is the loop of Henle. This loop of Henle, it's present in medulla region. After the loop of Henle, there are some more tunnels. These tunnels are called DCT, distal convoluted tubule. This DCT part of the nephron, it's present also in cortex region. This is the DCT. This is the DCT. This DCT part of the nephron, it's also present in cortex region. And there is another tube-like structure called collecting duct, which collects the urine. And this collecting duct, it's present in a medulla region. This is collecting duct. Here, this is collecting duct. As we know, the nephron is the functional unit of the kidney, which filtrates blood and produces the waste or produces the urine. So now we will talk about how actually the urine is formed, how the nephron filtrates the blood and produces urine and produces waste. So this is the Bowman's capsule. This is the Bowman's capsule. Here are space. There are space inside the Bowman's capsule. There are present space. That space is called Bowman's space. This space is called Bowman's space, where the glomerulus is present. Glomerulus is present in Bowman's space. Let me draw here the efferent and efferent. So let me remove it. This is the efferent arteriole. Okay, this is the efferent, and this is the efferent. Remember one thing, the efferent arteriole, this arteriole has more diameter than efferent. Okay, so in Bowman's space, here in Bowman's space, there are present bunch of capillaries. Okay, so the efferent arteriole comes and breaks in capillaries. Okay, like this. This is efferent. It breaks and many capillaries, and leaves it efferent. This is the efferent. Okay, the blood, this efferent arteriole brings the blood. This is, here are present a bunch of capillaries, that's called glomerulus. Okay, this is the glomerulus. By the help of glomerulus, the fluid is filtrated and produces the waste. So the efferent arteriole brings the blood. Okay, brings the blood inside the inside the glomerulus. The blood volume here, actually the efferent arteriole has more diameter than the efferent. The blood volume is high here. So when the blood comes and hits the glomerulus part, the blood comes and hits this part, this glomerulus part, and there creates actually the pressure, that's called the hydrostatic pressure. By the help of hydrostatic pressure, the blood filtrates. Okay, now when the blood comes and hits this glomerulus part, in glomerulus there are so many capillaries. The capillaries are fenestrated capillaries. The fenestrated means there are present some small holes. The holes diameter are about 30 to 100 nanometer. The holes, the fenestrated, which are present in capillaries, the holes are about 30 to 100 nanometer. So that particles which has size, those particles which has size 30 to 100 nanometer, they can pass easily. Okay, they can pass easily and comes in this space. So when the blood comes and hits the glomerulus part, the glomerulus has fenestrated. Fenestrated, there are present fenestrated capillary, there are present holes, and there are present so there creates the pressure. There are when blood comes and creates a pressure, that's called the hydrostatic pressure. After the blood hits this glomerulus part, the waste material comes in this capsular space. Okay, so those particles which has 30 to 100 nanometer size, they can pass easily. But when they pass, there are present some useful components. Okay, there are present also some useful components which we need to reabsorb. Now when the blood comes and hits this portion, hits this glomerulus, and waste comes now capsular space, the waste comes now in this part. This is called PCT, proximal convoluted tubule. So there is the another thing. There is when those things which have 70 to 100 nanometer. So those things which can easily pass here. There are proteins, there are present proteins like albumin protein. The albumin can pass easily from this hole, and the size of albumin is enough to pass from this capillary and from this portion and comes to the PCT or comes to the in this portion. But the albumin is a negative charged, and this glomerulus part, it's also called it's also negative charged, and they repel each other. That's why the albumin is not passed in this side. Okay, that's the cause. That's the cause why the albumin cannot pass the membrane. Why the albumin cannot comes in this side, because due to the albumin is negative charge, as well as this glomerulus is also, there are present base membrane, there are also present negative charge, so they repel each other. That's why the albumin comes not in this side. Now when the waste comes in this side, okay, the waste, what is the waste? There are not present blood RBCs, there are not present WBCs, there are not present proteins, there are not present platelets. So the left plasma which comes in this side, that's called primary urine, or we can also say deproteinized plasma, because there are not present protein. That's why we call it deproteinized plasma. Now when the particles comes, when the waste comes in at this side, there are present some important particles, and there are some useful components present. Now the useful components are like glucose, amino acids, sodium, chloride, or there are present water, so they need to reabsorb again. When the waste material comes now in this side, now the waste materials comes in PCT portion. This PCT portion is called proximal convoluted tubule. In PCT, the useful components are reabsorbed. Okay, for example, the glucose the glucose is present, the amino acids are present, the sodium chloride is present, or the water is present, which we need to reabsorb here in PCT portion. The PCT portion reabsorb these useful components. So the glucose is reabsorbed here. Okay, so the glucose is reabsorbed. Glucose is 100% reabsorbed here, and the amino acid is also 100% reabsorbed here. The sodium is 70% absorbed here, and the chlorine is 70%. These are 70% absorbed here, and the water is about 65% absorbed here. So these are the important components. So these are the useful components. They reabsorb again when they comes in this side, and PCT reabsorb these useful components. Now the filtration comes in this part. This is called loop of Henle. So the filtration, after the filtration, the filtrating part comes now in loop of Henle. The loop of Henle, there are two limbs. The first one is descending, the second one is ascending. The loop of Henle has some role in reabsorption. It reabsorbs sodium, chlorine, etc. But it is this descending loop. It is permeable for water. It reabsorbs water. Okay, this descending loop, it's permeable for water and impermeable for salts. But in descending loop, the remember here, the descending loop is permeable for water. Okay, it absorbs here water, and but this descending loop, it is impermeable for salts. And the ascending loop, this ascending loop is impermeable for water and permeable for salts. Okay, so the descending loop is permeable for water, and this is impermeable for water. The descending loop is impermeable for water, and the descending limb is impermeable for salts, and the ascending limb, it is permeable for salts. As now the filtrate comes in this portion. Here are present capillaries. Okay, around in nephron there are present capillaries. Okay, these capillaries are called peritubular capillaries. These are called peritubular because it's a tubular portion capillaries. The job of the peritubular capillaries is here when the filtrate comes, now when the waste comes here in DCT part. This is DCT part, distal convoluted tubule. Why we call it distal convoluted tubule? It's because it's for the glomerulus part. That's why it's called a distal. It's proximal because it's near the Bowman's capsule. After the filtration comes in DCT part. Okay, it comes in a DCT part where the secretion occurs. Where the DCT part also has some role in reabsorption. Sodium, chlorine, it reabsorbs here, but the main role of this DCT here, secretion occurs. Secretion, secretion occurs when the filtration occurs here. When the filtration during the filtration, some molecules or some particles escaped here, which are actually waste. The waste particles escaped here. In this filtration part, the waste material which escaped here, that is secreted here. Okay, that is secreted in DCT part. It is actually the DCT, the secretion is opposite to the reabsorption. Okay, in reabsorption there are some useful components reabsorbed, but in this part, in DCT part, the secretion occurs. In this secretion, those particles, those material which escaped, with those waste material which escaped in filtration part, those materials are pulled back to the waste, or to this DCT part. There are present peritubular. This peritubular capillaries. By the help of peritubular capillaries, the waste is pulled back to the this nephron. Like what type of waste? There are present ammonium present, there are present potassium, or there are present hydrogen ions, or there are present so many waste which escaped here. That waste pollute back to the DCT part, and that's called the secretion. Okay, so here are present peritubular capillaries. By the help of peritubular capillaries, the waste sends back. So when the waste comes back, when the waste sends back, and the waste comes now here in this tube. This tube is called collecting duct. It collects the waste. In collecting duct, the water is reabsorbed here. Okay, the water is there are present the ADH antidiuretic hormone. By the help of that, the water is reabsorbed here, and the concentrated urine is formed. Okay, here the concentrated urine is formed. So the filtration occurs, the reabsorption occurs, the secretion occurs. The three steps, actually there are present the three steps: the filtration and the reabsorption, reabsorb these important molecules, and the secretion which where the waste sends back to the nephron by the help of peritubular capillaries. So when the collecting duct, it collects the urine, and in collecting duct, the reabsorption of water occurs here, and the concentrated urine is formed. Now when the urine is forming, the urine is now collects the collecting duct. By the help of collecting, the urine comes now here. Okay, in previous video I talked about this portion. I talked about label about these all portions, like minor calyx. The urine comes now minor calyx. After the minor calyx, the urine comes in a major calyx. After the major calyx, the urine comes in renal pelvis. After the renal pelvis, the urine comes in a ureter. The ureters which are linked to the urinary bladder. The urinary bladder, by the help of the ureters, the urine goes down in the urinary bladder. Urinary bladder is a temporary state, that the urinary bladder, that is the temporary stage of the urine, where the urine stores temporarily, and by the help of a urethra, the urine is discharged out. So it was all about the formation of the urine. We talk about now some little more about this glomerular and this Bowman's capsule, this part of this Bowman's capsule. Here I made the visceral layer. And this is the parietal layer, parietal epithelial layer, which made actually the Bowman's capsule, and this is the PCT part. Okay, in this proximal connective tubule, these are podocytes, and this is the parietal layer, parietal epithelial layer, epithelial cells there, and these podocytes are linked each other by a special protein that's called the nephrin. These podocytes, they are present. These podocytes are attached to the basement membrane. They are present the membrane. The Bowman's capsule is made up of three things. Okay, and this portion, this Bowman's capsule, as well as the glomerulus, together they called renal corpuscle. Okay, they called renal corpuscle. So this is the Bowman's capsule. In Bowman's space there are present glomerulus. Together they called renal corpuscle. The renal corpuscle is made up of three things. Okay, made up of three things. The first one is, here are present capillaries, this glomerulus. There are present endothelial cells. endothelial, the capillaries are present, they represent endothelial cells. So beneath the, now after these capillary, they represent a basement membrane. The basement membrane is made up of collagen. Okay, it is made up of actually the collagen, and there are present podocytes, which actually the podocytes are linked to the basement membrane. The basement membrane, there are present these endothelial, so they represent capillaries. So in capillary there are present some small, these capillaries are fenestrated capillaries. So suppose these are fenestrated capillaries. There are present some small holes. By the help of these holes, the filtration occurs. Okay, and there are present some cells. Those cells are called mesangial cells. The mesangial cells produces lots of connective tissue. The mesangial cells acts as a phagocyte cells. So the mesangial cells produce connective tissue, and together the mesangial cells and the connective tissue together, they called mesangium. Okay, they called mesangium. So here represent endothelial cells, here represent podocytes. This is the visceral layer, and this is the parietal layer, which meets the Bowman's capsule. When the filtration occurs, there are present small pores, there are present small holes. The diameter of the holes are about 70 to 100 nanometer, and by the help of the fenestration, by the help of holes, the filtration occurs. So I hope you like this video. I hope you understand this video very better. If you have any question about this lecture, you can ask me in a comment box. I will try my best to reply you as soon as possible. So if you like this video, share this video, comment this video. Thank you.